right, so let's go ahead and get started. So good morning, everyone. Good morning. My name is Rita Blake. I am the manager of the Education and Outreach programs here at the California Nanosystems Institute at UCLA, aka CNSI. So welcome to CNSI. Just to give you guys a little bit of understanding, for those of you who have not been here yet, of where you are. So I'm sure you guys all looked this place up when you were looking at summer programs, but just to give you some introduction, so CNSI is a research institute, so we're not actually a department in the traditional sense. We don't grant degrees, but what we do is we facilitate interdisciplinary research, all centered around nanoscience. So we have all of the other science departments surrounding us, like life sciences, physical sciences, medicine, engineering, and they all come together at CNSI to do research centered around nanoscience. We also have an incubator that houses about 22 or 23 startup companies that are all centered around high technology, some of which, or at least a few of which, are also nanoscience based. And we also do a lot of different education and outreach programs, one of which is this summer program that your students all took over the course of this last week. So again, we do a lot. We facilitate research. We try to teach people about nanoscience and about all of the different skills surrounding it, and we try to incubate technology. And there's a lot of people that make both this institute and this program run. So there's a few acknowledgments that I want to give specifically for this program before we start. So first, we have to thank the leadership of CNSI for their continuing support of this program. Our director, Dr. Jeff Miller, who actually was also one of our guest lecturers for the program, so the students got to hear him speak on one of the big topics of his research, which was really lovely. And also our executive director, Sonia Luna, who helps us be sure that we're being fiscally responsible and pay the bills, but also, again, providing the means to support programs like this. We also had a lot of staff behind the scenes to make sure that the little things were ready, like lunch was ready, projectors were working, lab setups were ready, tables and chairs were ready, all of this other little things that, you know, are there and need to be ready to make a program run. So there's a lot of staff here that I need to thank. So thank you guys all, Elaine, Lucas, Nikki, Kevin, Mark. Thank you guys so much for all of your help in making all of this run too. And then, of course, the meat of this program, the nanoscience content of this program, and the microscopy content of this program, all centers around our faculty who are really wonderful researchers and leaders in their own fields. So our first instructor for this program was Dr. Adam Stieg, who's sitting right there, so you could say hello. And he is the associate director of all the technology centers that are here at CNSI. So of all the different labs that the students came in, Adam helps to oversee the operations of all of them and is also specifically the director of the Nano and Pico Characterization Lab. I'm sure at least some teams of which will have utilized some of his very cool technology and information to use. We also have Dr. Laurent Bentolila, who is also sitting right there, so you can say hello to him too. And he is the scientific director of the Advanced Light Microscopy and Spectroscopy Lab, and the scientific director of the Macroscale Imaging Lab. So a lot of the light and fluorescence and confocal microscopy techniques that students might also be using today for their presentations. We have Dr. Bentolila to thank for that because he really gave students a wealth of knowledge and hands-on experience in that area. And then finally, we have Dr. Hong Zhou, who's the director of the Electron Microscopy Labs here on campus and is also a professor in the Department of Microbiology, Immunology, and Molecular Genetics. I'm not sure if Hong has arrived yet, but we'll give him a nice applause anyway, and I'm sure he'll get here soon. And then we also had an extremely helpful and invaluable teaching assistant who is extremely competent in all three of the areas that our researchers, our instructors were leading and was very, very helpful in helping to guide students on their projects. And that is Michael Lake, who is right there. So we can thank him too. So just to give you all as parents a little bit of a perspective of what students did over the last week, because it was a lot, we had a very, very packed schedule, and students really got to get exposure to 3 different techniques in advanced microscopy. And then the goal for today was for them to work in teams and to utilize this knowledge of these different microscopy techniques to address grand challenges, which are initiatives that are identified by researchers and leaders in the government to really understand what are the big challenges that are coming up in science and what are the things that we still need to learn, what is the next generation of things we're going to be doing. And so the teams worked with these 3 different techniques that I'll talk a little bit more about, but then also looked at these techniques and how they could address either the microbiome challenge or the brain initiative. And I'm sure the students will tell you all about those, so I don't need to go into too much intro on that. But on the very first day, we sort of took them right into it. So they learned about nanoscience and nanoscale science by Dr. Stieg. They got a guest lecture about the microbiome from Dr. Jeff Miller and a guest lecture about the brain initiative by Dr. Katsushi Arisaka. And then they got to look at some of the different labs on the campus, like a clean room, a high-throughput screening facility, labs where they do animal imaging in vivo, so live animal imaging. And then they got to build their own microscopes and do some DIY microscope building. The next day, they learned about fluorescence microscopy from Dr. Bentalila, who also took them through a journal club to help them understand how to properly read a scientific paper, which can be daunting even for a graduate student. So to get the skills and how to read them as a high school student is extremely helpful. And then they also got an introduction into 3D modeling, so how they can take these images that they get and are processed through a computer, and how they can get them to be 3-dimensional images or models that they can really see and interact with. On Wednesday, they had an introduction to electron microscopy from Dr. Hong Zhou, and learned how to get even smaller images than light can get you. And then had some further looking into visualization and tools for computation and visualization with electron microscopy. And then yesterday, they finished out with our 3rd technique, which is scanning probe microscopy. And they learned how to see things not with light, not with electrons, but actually by just kind of touching them. And rounded out and finished out their work on image processing and 3D modeling. And then from yesterday until today, have been working on their final projects, where again, they're taking all of these current instruments and their understanding of them, and thinking about how they can take these techniques and use them to address real grand challenges. So these are not made-up ideas that we just come up with for the sake of this class. They're real, actual problems that scientists are looking at. So it was a lot of content in a very short period of time, and I'm sure that we're all going to be extremely impressed by what high school students can come up with in 5 days. So with that, I'm going to stop talking and let our students do the talking. So I will call up group 1. So team 1, do you guys know who you are? Darwin, Kyle, and Jonathan? All right, well, come on up. Let's give them a round of applause. All right, so Mark, could you help me find their... I can't seem to locate where they are on the drive.
What did you call your...
Looks like... Group 1. Mm-hmm. It's on the wrong screen now.
guys. We are the saviors of the soil. My name is Darwin Kim. I go to LaSalle High School in Pasadena.
I'm Kyle Herkins. I go to Laguna Beach High School down in Orange County.
I'm Jonathan Hung. I go to Shadyside Academy in Pittsburgh.
Right, so in abstract, as we all know, plants and microbes interact extensively through a symbiotic relationship. These communities of microorganisms, also known as microbes in the environment, live in and outside of plants. These microbes serve various functions such as decomposing and recycling old plant matter and providing plants with nitrogen and phosphorus, which is important for their chlorophyll, which is, of course, where photosynthesis takes place. So yeah, in this way, microbes indirectly affect us humans as well because we're the ones farming, appreciating the plant's beauty, and even eating the plants. So our scientific question is, how can we make plants and microbes safe from major metal pollutants such as zinc, copper, and lead? Our solution and approach would be to genetically engineer the microbes present in the soil, specifically Streptomyces griseus, which is a generic species found in almost all of the farming soil. We plan to edit the genome of these microbes and of these bacteria to make them more resistant to pollutants, using a gene that codes for resistance to these heavy metals, which would be purified from bacteria called C. metallidurans, which is often found in biofilm and in wastewater outside of industrial factories with high concentrations of heavy metals. So yeah, this question is significant because the symbiosis of plants and its microbes is currently endangered by certain threats such as climate change, soil degradation, and poor land management practices. Of course, we can't solve all of these problems, but we can try to eliminate at least one, therefore, and make a pretty big impact. And potential impacts is sustainable plants and microbes, and perhaps once our lab work is established, the global distribution of these genetically modified bacteria in the form of probiotics or maybe even in fertilizer. All right, so in concept, our question again is, how do we better protect plants and its microbes and its symbiotic relationship? And our mission is to preserve this harmony and address a problem, which is heavy metal pollution from runoffs and other things. So yeah, this topic is totally relevant to what we were learning because this is a cycle and it affects us humans as well. And it directly concerns the microbiome.
Thanks. Okay, so all the science stuff is great and all, but the important thing is, how does this affect everyone? How does this affect the global community? The soil that we're looking into is the kind of soil that would grow on farms, or the kind of soil that would grow our food. So if this soil has these pollutants in them, those pollutants can eventually find their way into our foods and eventually us. And personally, I don't really want to eat polluted food. So by seeing what we can do about the pollutants in the soil, we can potentially make our food even safer to consume. Also, by looking around in the soil microbiome, we can possibly find other problems that could get into our food and attempt to solve those as well. Next slide. Thank you. Pollutants in soil is not a very new topic that people have been looking into. There are many studies that have been looking into how the soil is affected by these pollutants. However, very few of these studies are actually looking at how the soil microbiome is affected by and affects these pollutants. So that's what makes our study very special. We are actually taking it down to that very small scale to see how things down there affect the big picture with the soil. Yes.
All right, so the first step in our team's approach would to be we need to find a bacteria that has desirable traits that we'd want in the plant microbes. So the one we found was C. metallidurans, and this bacteria is resistant to heavy metals and is able to ingest these heavy metals and make a non-phototoxic byproduct, which just means these byproducts won't harm the plants. So after we extract the DNA, we're going to try to cut out the DNA responsible for these traits and replicate them using PCR. So just PCR, in the first step, you would heat up the mixture to break the double bond, the double helix, I mean, and then you would cool off the mixture and add primers so that they can attach and add nucleotides. And then you would heat the mixture up again, and the nucleotides would add to the primers and extend, and then you would have more DNA. And then you just repeat these steps until you have a bunch of replicated DNA. So now that we have our PCR product, we want to get it into the plasmid of plant microbes. So we would take the plasmid out and cut it with restriction enzyme, and then we would try to ligate the plasmid in our PCR product. And then after we have these plasmids containing this DNA code, we would heat shock it back into the plant microbes so that hopefully they can express these traits. So to check to see if these plant microbes actually took up the plasmid, we'll grow them in a dish with heavy metals present so that only the plant microbes with the heavy metal resistance will be able to survive. And then to see if they have the other trait, which is we want them to be able to ingest these heavy metals and also create a non-toxic byproduct. So to check that, we'll look at these bacteria under a fluorescence microscope and see if they're actually doing this process. So our greatest challenge is finding bacteria with these traits so that we can put them into plant microbes. So for our example, it's C. metallidurans. And this bacteria is really important because it not only is resistant to these heavy metals, but it's also able to convert these heavy metals and toxins into a byproduct where the plants aren't able to consume it. So we don't want to be eating plants with these toxins in it. So to sum it up, we're looking for bacteria that have these desirable traits that we'd want to put into the plant microbes. We're going to take the DNA out and replicate it using PCR, and then we're going to ligate it into the plasmids of plant microbes, and hopefully they'll display these traits.
Okay, so these are our team members here, and we are done presenting. So thanks for listening. So we have time for some questions. So if you have a question, go ahead and ask. No questions?
All right.
Okay.
So one issue is your plan is to take microbes that are universally available in plant soil and make them able to convert these metals into a non-toxic byproduct.
Yes.
Right? So in this case, first of all, let me compliment you on the level of detail in terms of being able to actually plan this genetic strategy to basically transfer these genes and see if you can develop this new property in your resulting microbes. I think that's actually a very, very good detailed explanation of how you would approach that. But the question would be then, after you find this bacteria and you spread them out into the wild, how are you going to make sure that they thrive? Right?
So what we're planning to do, with the bacteria, we're going to start small. We're going to keep it to like maybe farm areas. We're not going to try to expand it all around the world. And we'll have to have a pretty good sample. Otherwise, as you said, if we only put in only a few of the modified bacteria, they might die, and then we'd have to start back from square one. So, yeah, so we'd start small, we'd see how it goes, and we'd make modifications as we go from there.
And maybe we could add on some sort of like genetic code or like advantage to give them a step up over the like wild or natural microbes so they would thrive.
On a farm like
Yeah.
pesticide resistance or something like that.
Yeah.
You have to look at how that affects microbes. But
Yeah.
interesting ideas.
Okay. Yes?
So you chose fluorescence microscopy from the 3 techniques you learned.
Yeah.
In terms of the proposal you have, which I agree with Mike, is quite nice, what is the key insight you would gain from using that technique about your proposal with respect to either visualization of these microbes or the actual output of this
Right, so from the various techniques that we learned this week, obviously it's a biological sample, so we don't want to kill it using electrons. But I think since we're dealing with bacteria here, we only need to look at the cellular level, and we need to make sure if it's producing the non-toxic byproducts. So any typical light microscope would do, and we thought that fluorescence would do the job.
So I guess I'll take it further. So what would you hope to see from the fluorescence, right? Because the images that you showed, my understanding would be you would have to fluorescently tag some element that showed that you're basically insertion of this DNA has occurred, right? So you know that you've manipulated these bacteria. But then how do you know that they actually are doing what you think they're doing?
Right.
Using microscopy.
Right.
There are other readouts that you could use.
Right, so under the microscope, in the presence of these heavy metals in aqueous solutions, to make sure that it worked and that the bacteria have the desired trait, we would be able to see the byproduct, which is solid metals and solid copper, solid lead, things like that. So, which is, of course, which is hard for plants to digest. Yeah, so that's what we'd be looking for solid metals. Yeah, I think we would be able to see that under a microscope.
Is there one more question?
Students?
So if we don't have any more questions, then we're done here. So thanks for listening again. Good job, guys.
All right, so team 2, perfect.
So while they're setting up, I will say, students, we would like you to ask questions, right? This isn't just going to be us asking questions. Part of a vibrant community of learning is to ask questions to your peers. Right? This isn't meant to be a test where the instructors are over here asking all the questions. Right? So please think about if you have anything that strikes you as a potential question. Formulate something. We'd like to have questions from the students as much as possible. And the parents are also very welcome to ask questions.
Even if you don't understand something, feel free. These students would be happy to explain it to you.
So hi everyone, my name is Katie Kayan.
And I'm Sarah Ramirez.
And I'm Anna Hashimoto.
And so the research question that we will be answering with our proposal is, how can the study of the degeneration of neurons in Alzheimer's disease be used to stop neuronal damage?
So, the degeneration of neurons can be studied using cryo-electron microscopy and fluorescence microscopy. Our technique can be practiced in labs and tested and of use to scientists. The how and why neuronal damage is still somewhat unknown, and therefore, sadly, there's no cure for Alzheimer's. The disease is troublesome for patients and families all around the world. Therefore, we search for answers to progress the Brain Initiative and discover more about neuronal damage. So what is Alzheimer's disease? It is a condition that impairs memory, thinking, and behavior, and it's caused by the buildup of proteins called beta amyloids, and they form plaque in the brain, making it difficult to think clearly and remember things. There's also another protein called the tau protein that causes tangling and also messes with thinking processes. This leads to degenerative neurons, and therefore the brain is shrinking because it's made of neurons. Yeah. Alzheimer's is a type of dementia. There are 3 broad stages of Alzheimer's. There's preclinical, which would be before many symptoms like forgetting, and there's mild cognitive impairment where the Alzheimer's develops, and then you start experiencing symptoms, and ultimately dementia. And like I said before, there's no cure.
So just to give you guys a little background about what prions are a type of microbe and they stand for proteinaceous infectious particles. And like its name suggests, prions are proteins. However, there are two forms. One is the misfolded form and the other one is the normal kind. And the misfolded kind can cause the normal prion proteins to develop into misfolded ones by causing a deformation in a region known as alpha helix 1. And so the prion that is specifically involved in Alzheimer's disease, which is what we're looking at, is called beta amyloid protein. And this beta amyloid protein accumulates in the brain, forming plaques, like Sarah said.
So as of last year, nearly 44 million people around the world had Alzheimer's, and it's common. There was also a study that said 30% of people with Alzheimer's also have heart disease, and 29 also have diabetes. So we know that Alzheimer's causes the degeneration of neurons, and therefore scientists are, and with using our techniques, we're trying to stop the progression of neuronal damage before it occurs and spreads, to inhibit like further damage that can worsen someone's condition.
Okay, so I'm going to be talking about some of the studies that have been done by scientists and things that they found to try to advance the cure for Alzheimer's. So one of these is a study that was published in Nature magazine just last year, and it was talking about how scientists were able to discover the structure of the tau protein. And the tau protein is one of the proteins in the brain that is one of the major causes of Alzheimer's because it tangles inside the brain. And so they were able to study the structure of the tau protein using cryo-electron microscopy, and they're hoping to develop new drugs that could perhaps target this tau protein and destroy it. However, one of the setbacks to this approach was the fact that the tau protein is involved in creating microtubules, which transport materials within the brain, and as a result, destroying these tau proteins could have some unintended consequences. Another one of the studies that we looked at was the discovery of the role of exosomes in spreading Alzheimer's disease. So exosomes, they basically transmit information inside the brain, and they contain genes and proteins that can be absorbed by other cells. And so in one of the studies, the scientists purified the exosomes, which still contain the beta amyloid protein, and they found out that these exosomes could still be absorbed by normal healthy cells, meaning that the Alzheimer's disease, the beta amyloid protein, had spread to other cells. Another one of the studies was the connection between viruses and Alzheimer's. So there have been research has shown that the level of the HIV virus in people with Alzheimer's is twice as high as in normal people. And however, scientists don't know for sure whether this link between HIV and Alzheimer's is correct, but there is interest in creating antivirals as a potential cure. And the last study that we looked at was the depletion of an enzyme called BACE1. This enzyme produces the beta amyloid protein that is one of the major causes of Alzheimer's. And so scientists reversed the formation of this enzyme, and as a result, were able to halt the progression of Alzheimer's. And however, this approach has only been tested in mice, so scientists are unsure if it will work in humans as well. So for our project, we plan to study the degeneration of neurons in the brain in order to help researchers better understand the degeneration of neurons and perhaps create a cure that would stop neuronal damage. So we plan to do this using two types of microscopy techniques. One is cryo-electron microscopy, and the other one is fluorescence microscopy. For cryo-electron microscopy, we're going to view slices of brain from mice in order to better understand how neuronal damage affects the brain as a whole. And the second follow-up technique to this would be fluorescence microscopy, and this would be our main approach, which would to be able to study the activity of the neurons and monitor their progression. So we plan to insert exosomes into the brains of mice in order to give them Alzheimer's so that we'll be able to monitor the progression of the disease from the very beginning to the end. And we plan to create a fluorescent dye that would target calcium because when neurons fire, they release calcium. And in order to monitor this neuronal activity, we plan to use this fluorescent dye to be able to see when the neurons slowly stop firing and die off. And secondly, we plan to be able to design another fluorophore, which would target the prion proteins inside the brain, or the PRPSC, which has membrane glycoproteins that would allow the fluorophore to attach to it. And in doing so, scientists would be able to better view the prion protein and understand it.
Okay, so we have proposed to use Cryo-EM to observe the degeneration of neurons in mice brain samples. However, because Cryo-EM involves freezing the samples before viewing it, we will need to obtain multiple samples of mice brain slices from all stages of Alzheimer's. And that is both very inconvenient and troublesome. Another challenge we face is that when we monitor the full effect of Alzheimer's through fluorescence microscopy, we will need to begin from the first stages of Alzheimer's to the very last stage. And this process is very time-consuming and is a lot of data to process. To summarize, we propose to utilize Cryo-EM to view the brain slice samples of mice affected with Alzheimer's. And through this data, we will be able to understand how the degeneration of neurons affect the brain at different stages of Alzheimer's. We will also plan to create a fluorophore that would attach to the calcium-2+ emitted by the neurons in the brain. Through fluorescence microscopy, we will be able to view the fluorophores, which the amount of fluorophores, which actually correlates to the activity of the neurons, and therefore we can observe the degeneration of the neurons through the changes in the amount of fluorophore. We will also create another fluorophore that will target the PRPSC, or also known as the cellular prion protein, and bind it to bind to it to affect the neurons with Alzheimer's disease and make it fluoresce. And the combinations of the two techniques will enable us to study the structure and monitor the degeneration of the neurons. And through the two techniques, we hope to collect information on the activity of the neurons and how they deteriorate through Alzheimer's. And we hope to provide researchers, through our data, we hope to provide researchers with information on how they may be able to find a cure for Alzheimer's in the early stages of Alzheimer's.
So here are our contributions and our references. So thank you. If you have any questions.
Yes.
I'm curious as to if studying the neurons of mice may be more beneficial than studying the neurons of other animals.
So we chose to study mice because mice have been used in other research to study Alzheimer's, and also because mice often resemble humans, and also because of the ethical considerations of using larger animals. For example, mice, they're smaller and easier to access and more convenient to use. And also by putting Alzheimer's or genetically engineering the mice to have Alzheimer's, they have been shown to show the same symptoms as humans would have if they had Alzheimer's disease.
Any more questions? Yes.
So to go off on the previous question, do you think it will be safe to infer the data from the tests of the mice to actually use that data to treat humans? And maybe because they're completely different. Well, not completely, but they are different things, and you can't really just use data from a mice to treat a human, and because you can't really test a human, you can't really give a human Alzheimer's. So, do you think it will be safe to use the data?
Okay.
So I think when the ultimate goal is to eventually test it on humans, so I'd say mice first, and then once we gather enough information and we're sure how to apply it or convert it onto a way that's safe for humans, of course it'll be tested first. Like some people, they volunteer for these things, or they need like something right away, so they can try dangerous new methods. But ultimately the goal is to use this method on humans eventually. So we won't do it unless we know it's for the most part safe.
And also a lot of researchers, they've been studying Alzheimer's through mice. And this is because, like I said earlier, the mice, when they are given Alzheimer's, they show more symptoms that are related to humans if they had Alzheimer's too.
They also have similar structure, like their brain, their organs, so that's why we use mice. Yes?
What do you hope
So currently scientists, they don't understand, or they know that neurons degenerate with Alzheimer's. However, they're trying to find a cure in order to stop this damage from occurring before it starts. So we plan to use our research to be able to help scientists better understand how these neurons degenerate through Alzheimer's and perhaps monitor their progress and activity. Yeah.
So we have one more question from Dr. Ventililo.
It's a very elegant proposal, and that what I really like is that you actually brought in two techniques to try to address a research problem, which is really nice. So if I understand correctly, you said you're going to use brain slices, so kind of a fixed sample to use on electron microscopy, and then more live imaging, right, using fluorescence, which is all makes also a lot of sense. And I wanted to ask you, do you think it would be possible to actually use both techniques almost at the same time? Maybe not at the same time, but maybe one after another on the same type of samples, and what would you learn from that?
Yes, I think it would be possible, but for the cryo-electron microscopy, the brain samples, they would have to be frozen. So for the fluorescence microscopy, we plan to test it on live mice, and then perhaps after the disease has progressed and towards the end stages, we could use the brain samples from the mice that had the Alzheimer's and test it under the cryo-electron microscopy to see the effect of how the damaged neurons affected the entire brain.
Right, so it takes the same samples basically and get them first with the fluorescence microscope and then very good.
Yes.
All right, thank you, ladies.
Thank you.
Thanks.
All right, so group 3.
Should name something different.
It's this one.
This one?
Yeah.
Yeah.
Okay, hello, my name is Rebecca Barrick and I go to Moore Park High School.
I'm Kelly Sue and I go to Temple City High.
I'm Ria Kumar and I go to Northwood High School.
And this is our presentation on how the microbiome and stem cells interact.
So for our abstract, just to summarize it, we chose to focus on the gut microbiome, but specifically on intestinal stem cells. These stem cells are often used to replace epithelial cells, which are the cells that exist in the inner lining of your stomach, when they shed periodically, as well as in case of any injuries. In the past, antibiotics, probiotics, and bacterial strains have been found to impact the gut microbiome, but not much research has been done on the intestinal stem cells themselves. So using our results, we'd like to use the research to help stem cell deficiency in the future.
So the main question that we're trying to answer is, how do probiotics, antibiotics, and bacterial strains that affect the body's microbiome affect stem cell populations? So there has been previous research that shows that antibiotics and similar products affect the microbiome within humans in a negative manner. However, not much research has been done on how the negative effects on the microbiome will affect stem cell populations, since stem cells are very important to the development of humans and the regeneration of cells in general. So stem cells are an essential part of human development. However, there is a limited amount of them. We currently are able to find and cultivate these precious cells, which we are currently using to develop new types of drugs to treat different diseases. We know that the microbiome is negatively affected by antibiotics, which are currently necessary to treat bacterial infections. It is currently under question as to if stem cells are affected by the human microbiome, and if so, how. If stem cells are harmed by antibiotics, which destroy the gut microbiome and can cause dysbiosis, or aided by probiotics, which help promote the gut's natural flora, then this could be indicative that the gut microbiome has an effect on stem cell health. If this is shown likely by the data, this would change how we treat patients with stem cell deficiencies and how we treat diseases in relation to a possible impact on stem cell vitality. So the issue also affects us daily in California. We constantly hear news articles about how restaurants like Chipotle or crops such as lettuce have experienced outbreaks of E. coli. a pathogenic bacteria. If it is found that these pathogens do affect stem cells, we could be having them reduced daily due to these outbreaks.
Currently, researchers are establishing background information by looking at the normal functioning of intestinal stem cells and what they do, like when they're behaving normally. They normally replace epithelial stem cells when they decompose, like periodically, as well as when you get injured. There has also been research on cell markers and what they indicate for the intestine. Understanding the function of these stem cells is vital to further carrying out the research on how they can be impacted by the antibiotics, probiotics, and bacterial strains. But another example of how such results have been carried out in a different stem cell location is antibiotics that have been tested with the adipose-derived cells, which are stem cells that exist in your fat. And they have shown that administering penicillin, which normally treats infections like your ear infections, have been shown to negatively impact cell differentiation. This information is vital to guiding research in the intestine and how we can use it to help the future.
So our proposal is to study the effect various conditions in the gut microbiome have as an effect on stem cell production. This would include studying the effect of antibiotics, which are known to greatly reduce the amounts of both healthy and not so healthy bacteria in the gut microbiome. Probiotics, which are intended to restore healthy bacteria, such as lactobacillus, to the microbiome, and also studying the effect of certain pathogenic strains on the microbiome, such as salmonella. Our model organism would be rats, who we've treated with the aforementioned conditions. We would then study their stool samples to assess the bacterial populations within the microbiome, and then extract stem cells from the rats by tagging them with fluorescence proteins. The extracted and tagged stem cells would then be assessed using confocal microscopy to view their abundance and ability to divide. We also could use scanning probe microscopy to examine them, because I found a diagram in a paper discussing how they can sense different types of cells using scanning probe microscopy, and so we could assess the health of the cells based on the information collected by the scanning probe microscopy. So some potential limitations of this project that we may encounter include the large amount of time it would take to test both the effects of antibiotics, probiotics, and pathogenic bacterial strains on rats, and then also having a control group to make sure that our data is viable. Another limitation that we could encounter would include the amount of money and time that would be spent gathering data from the gut microbiome and the stem cells, and then using it to study on the microscope. So discussion. So overall, this project would seek to define the relationship between the gut microbiota and the stem cell health through stimulation of different gut microbiome conditions in rat subjects, and then studying the samples using fluorescence microscopy. A future direction that this project may follow would be looking at treatments for stem cell deficiency and establishing the best conditions for stem cell prosperity. We could also look at different types of microbiomes, so maybe the skin microbiome and how that affects different types of health, or how the gut microbiome affects the nervous system as well, and different stem cell systems.
So these are our contributions, and these are our references, and thank you.
Questions?
Yeah.
Yeah?
How do you plan to get stem cells?
So we focused a lot in the research and the lit review, we saw a lot about intestinal stem cells, and so we would extract the intestinal stem cells from the mice and then look at them underneath the microscopes and judge whether they're being produced a lot, or if like with the bacterial strains, what we would predict would be that they'd be reduced because bacterial strains might affect that. And so if there's like a decreased amount of stem cell growth, then we could determine how the microbiome affects their growth and production, and whether they're healthy and able to divide or not.
So we could also get a sample of where we think stem cells are located, and then we could stain the sample to just single out the stem cell, and just cut that sample off, and then the stem cell off, and let it propagate.
Other questions? Yeah, Michael?
When you're talking about tagging the cells, are you talking about extracting the cells from the mouse and then trying to use the GFP to identify the stem cells? Or are you talking about trying to put a genetic marker in the cells so that the stem cells naturally glow green and can just be counted?
I was thinking putting in a genetic marker, because when we were in the lab with Professor Bentalilo, we saw the microdissection machine, and he mentioned how he used the GFP to be able to extract those from the bones a lot easier. And so that's what we were thinking along with the fluorescence.
So then you could quantitate histology slices by how many green cells are in them.
Yes.
As long as you count enough of them, it should be statistically representative of how many stem cells were present in the tissue.
Yes.
Great.
Another question?
I'll just make one quick comment.
Yeah.
I really like the fact that this is very novel, right? And that you're looking at these connections, but it's very dangerous in science to just go shoot in the dark and say, oh,
Yeah.
these things have no data behind them. So the fact that you were able to find a case like the adipose tissue, where you actually have seen an effect like this, you don't want to be, it's great to be the first one to discover something, but it's also very risky territory. It usually comes from observations in other projects that those discoveries are made. And so the idea that you have a defined effect that you know exists, at least for other tissues, and wanted to see if there's an effect on this other tissue, I think gives you a light in the dark, so to speak, to go ahead and approach this. So I think that's an important aspect when you go search. Otherwise, you have to worry about it being a fishing expedition, is what people would accuse you of in grant proposals.
Oh, yeah. Oh, two more questions.
Yeah, please.
Okay.
So, if I remember correctly, are there other parts of the body where you can find stem cells?
Yeah, they're in like bone marrow and then adipose
also-
cells.
So, why are you is there a reason why you're specifically choosing to look at the gut microbiome and the gut stem cells?
So from the pre-reading, we read a lot about the Microbiome Initiative and how it's focusing a lot on the gut microbiome. And when we were doing a literature review, we did find stuff on adipose cells, but we also found a lot on intestinal stem cells. And so that's kind of where it went, because a lot of information, it was like in 2016 that this study was done, and they were defining how epithelial cells interact with stem cells in the intestine. And so that's why our project took that direction.
I was just going to ask, I know you heard from Professor Miller talking about not just what's in the gut microbiota, right, but where it's located. Right? The fact that you can extract out the way that you're talking about now and isolate everything and measure it and quantify it. But the fact that they actually have locations inside the intestine matters, right, and there's sort of spatial distribution. So I'm wondering if there was anything you learned this week that might make you think you would be able to do this in a live organism instead of having to extract out the cells. Did you hear anything this week, or is there any possibility you could imagine where the fluorescence technique could be used in a living rat
Oh,
instead of
yeah.
having to isolate the cells from the stool
So-
or extract them?
So there are PET scans that can be used. So if we were to, I noticed in the lab that they like injected it through the tail, and they'd have different markers that would attach to different parts. So if they attach to the stem cells, or different, if we wanted to look for different strains of bacteria in the microbiome in the gut, we could see those using the PET CT scans. Thank you.
Thanks.
Sarah.
Yeah, I'll just make a quick note. So we will take a break in the middle of presentations, but if you guys feel an urge to use the restroom, please feel free. They are just down the hallway, down that way. So when you exit this auditorium, just go down the hall past where the offices are and you'll see signs.
The good thing about a college course, you don't have to ask if you can.
So which one is?
Parker. It's on Parker. Wait, I think it's this one, right?
This one? Yes? Okay.
Okay. Hi, I'm Parker.
I'm Tianjun.
Hi, I'm Daria.
All right, and today we are going to be talking to you guys a bit about tagging myelins with green fluorescent proteins inside of the brain of patients with demyelinating and degenerative brain diseases. Okay, so let's see. Myelin is a protein that forms inside of the brains of most living organisms around the nerve cells, specifically the axons, which are the main communication systems between each and every neuron inside of the brain. And it's essentially what creates a functioning circuit which delivers electrons. And in order to move these electrons as effectively as possible, say when you are practicing like a dance move or recalling information, these electrons will travel through the axons, and as you become better and better at doing such things, the more protein will be established and grown around the axons, which is essentially this insulating protein. It's an insulator that helps retain as many electrons and keep the flow going as strongly as possible. However, there are many diseases and many neuronal conditions which can unfortunately break down and stop the processes that help create myelins around the axons, which including Parkinson's or multiple sclerosis. And this demyelinating process can cause motor issues, issues regarding the retrieval of memories, and it can be generally bad for the sufferer or patient. And so we believe that we can actually next. So our concept is to tag these neurons with green fluorescent proteins within their own genetic code, and to essentially try to locate and study each individual neuron over a set period of time, and also being able to study it under certain conditions, while also being able to observe and image it through different methods of fluorescence microscopy and spectroscopy, while the patient or subject specimen is still alive. And you wouldn't really need to remove any brain cells necessarily to observe them and how they are affected by these diseases. Okay, so, well, many people are affected by these diseases, such as Parkinson's and multiple sclerosis, and, every year there are, thousands of people who are diagnosed, and generally that is devastating news, and it can, it's just generally very bad. And as it continues to spread and attack the brain, it can cause degradation of memory and behavior. Okay, so, but we believe that by, again, tagging the genetic code of the neurons with green fluorescent proteins, we can hopefully better understand the complexity of how the brain reacts to these diseases and how it's affected. And we believe that these studies could help us hopefully one day create a cure or prevention method from actually getting these diseases.
So common studies have been done on zebrafish and mice. So these experiments were conducted 6 months ago, and they were exposed to a myelin-related disease called demyelination, as he mentioned earlier. And scientists studied the effect of the GFP-tagged myelin and in these organisms and found that the diseases were easier to see, and they could understand neurological concepts and mapping the brain in an easier way. Okay. So our proposed project is to essentially tag the genetic code of neurons in the brains of the demyelinating diseases suffering patients with green fluorescent proteins. By using GFP tagged myelin, we can have the ability to trap individual neurons and study them over a certain period and examine the brain for further details that might help us determine how the disease is caused and how it affects our brains, and find ways to prevent and cure myelin-related diseases.
So along with our proposal, there are potential challenges that will arise that we have to take into account. Well, since the GFP is essentially a guide, and since they guide by providing a visible glow, and it will be really difficult to monitor the GFP inside like a deep inside the tissue of a brain. And to do that, well, there are a variety of ways to actually do the imaging. Well, what we've learned over the course of these days are there are the scanning probe microscopy, electron microscopy, fluorescence microscopy. But then, even though there's these new and improved methods of imaging, it is still really difficult to see inside the deep tissue of the brain, because if we can do that, then technically we wouldn't be here. And also, since we have to introduce the modified nerve cells into the affected victims of the disease, we have to technically extract some healthy cells from them to actually avoid collisions. Well, not collisions of the modified cells. We have to prevent the potential invasion. So this method will be really personal, based on the patient himself or herself. So we can't mass produce this potential cure. And so overall, the GFP-tagged myelin proteins could possibly allow many scientists to study the complexity of the neurological systems and to develop treatments and cures to brain-related diseases with fluorescent microscopy. And with this discovery, we can look at the brain diseases on a whole new viewpoint, and the potential future, in the near future, we can potentially be able to track back the diseased brain cells to find the origin of the disease and potentially to solve the problem. And here are the contributions and references.
Any questions? Yeah, any questions?
One question, in the idea of removing nerve cells, what would be one of the major problems you would encounter? You're trying to treat a patient whose nerves are suffering from a disease. So in removing those cells, what problems you would encounter?
Okay, so, well, the thing about these nerve cells, these neuronal cells inside the brain, as they're being attacked by these degenerative diseases, there is a very high risk of them ultimately being destroyed or killed off by said disease. And of course, there's also ways that these cells will, not be able to fully connect and be integrated into the rest of the brain. And, that's because the nerve cells, they need to be connected by the axons. However, there are lots of different methods being created to fuse cells, nerve cells, neuronal cells together to one another in order to create an established connection. But, you know, again, as the cells are being attacked, it's very risky, and, you know, for said cells. That's why you need to find a lot of them, and you need to find a lot of younger cells that might also be healthier, perhaps say from, like, you know, a donor or a stem cell, you know, donor that could potentially help grow these brain cells. There's also lots of other methods of creating brain cells through stem cells in labs, or essentially mini brains, as they call them. But these stem cells would essentially, again, be tagged with a GFP so that we can track them as they divide throughout the brain, or probably, well, this way we can actually track specific neurons while also trying to
find the causes of the diseases.
Yeah.
So let's say if you take the neurons out, you're going to immediately cause the damage that you're trying to prevent.
Exactly.
Right?
Yeah.
However, if you're really lucky, are you aware of which type of cells actually make myelin? So it turns out it's a neuronal cell,
yeah.
it's not neurons.
Yeah.
It's glial cells.
Yeah.
And it turns out
the
that
glial cells.
you guys are in one of the few areas.
Mhm.
They are removing glial cells from patients, growing them up in batch cultures,
Yep.
and re-injecting them into patients right now. So it sounds like it's sci-fi that you would ever be able to do this.
Yeah.
And you can't do it with neurons. That's
Exactly. Yeah. No.
out.
You. Yeah.
Right? But you could add a GFP tag to those. So then the next question I have is, what are you going to learn by studying humans that you haven't already been able to learn from rodents and mice and these other models?
Well, let's see. So.
Well, maybe we'll have the kind of a safeguard that, you know, this worked on humans. It worked on one, maybe it'll work on the others. And, you know, with a mouse or rodent, it technically is, it could create some trusting issues.
So they're trying to validate the research from mice.
Yeah, potentially.
And, you know, humans have much larger brains than mice, and so there's lots of different areas of the brain for which these different diseases could attack and target specifically. I think that by studying how it works in larger brains of larger organisms, we could better study exactly how and why this disease does, you know, what it does, and this, you know, in these different areas of the brain. And I think that although mice, you know, are a good test subject to use for these methods of disease prevention or repair, human brains, you know, they could also provide a much larger area to work with and to better understand exactly how these diseases, you know, can affect a brain based upon exactly where, you know, it, again, does pinpoint and focus its areas of, you know, damage. Like where these, yeah, you get it. Yeah.
So I think we have time for one more question. I think, Dr. Steve, you had a question?
Yeah, I was just going to ask, you mentioned it, I mean, this point here on the top, I'm going to just ask you again, similar to the last group. Did you hear anything this week that would allow you to see deeper into the brain? Right? I mean, I know that you did, so I'm just wondering if you thought about other methods that would allow you to basically look at demyelination using fluorescence microscopy throughout a brain rather than by extracting cells.
Yeah.
And what the limitations of those approaches might be.
Yes. Well, again, there are many different types of fluorescent methods through microscopy. There's, of course, removing brain samples or tissue samples that have been tagged with the GFP or other fluorophore proteins and markers. And, that's great and all. You can see the individual cells up close and in a very clear image. However, of course, through doing that, you can either end up damaging the live specimen or injuring it or ultimately killing it if you are removing brain cells. But there are other methods of microscopy, fluorescence microscopy, that can be used to observe the live samples or specimens without actually causing damage to them, such as, if I'm correct, MS spectroscopy, which is well, yeah. There's many types of scanning machines which can essentially highlight and observe the fluorescent reactive proteins or materials that can be found within a live specimen fully through methods of scanning. We talked about it a little bit while we were doing the tours and talking about methods of fluorescence microscopy and using live mice, specifically more or less ones that have had quantum dots injected into their bodies. But of course, it's also possible to use other types of fluorophores that might be more, might be easier to work with and easier to observe.
So you are group 5?
Yeah.
Perfect. Pardon me?
Hi, we're doing our presentation on neural circuits, and I'm Audrey.
I'm Francis.
I'm Aideen.
So essentially, our abstract is that because neurological diseases and psychiatric diseases are two of the most common diseases worldwide, types of diseases worldwide, and a lot of these diseases don't have cures or better treatment yet, we would like to study the complexity of the brain and its neurological circuits. So because of the brain's complexity, a lot of scientists have difficulty finding these cures for a lot of diseases such as Parkinson's and Alzheimer's. So in order to better the research done, we would like to use fluorescent microscopy to better picture these neurons and to fully map out the neural circuits, which are networks of interconnected neurons that impact behaviors. So with better research done on these neural circuits, we could potentially better treatment done on current diseases as well as be more prepared for future diseases that may arise.
The main question we will be addressing is how can we use fluorescence microscopy to better understand neural circuits for whole brain mapping? So the brain is one of the most complex and least understood organs, and 1 in 6 people have neurological diseases, and 1 in 4 people suffer from psychiatric diseases. Many of them currently classified or deemed as incurable, like Alzheimer's, Parkinson's, dementia, which can cause people to be bedridden, in wheelchairs, lose cognitive function or mobility. And through understanding more about the brain and its neurons, we hope to gain insight in not just one disease, but many diseases. And hopefully through this insight, create cures for the diseases, and this will help many people to take back their lives. and those affected will also be able to live with less pain and discomfort, hopefully. And for the more complex diseases, we'll hopefully be able to provide like some help, and not necessarily a cure, but help them.
So currently there are many different ways to study the brain. So firstly, functional magnetic resonance imaging. It focuses on using strong magnetic fields and radio waves to create detailed images of the body, and it also researches the blood flow in the brain. So areas that have more blood flow than others are studied, they show that they're used more often than others. So transcranial magnetic stimulation. So what this is you have a magnetic coil on the scalp, and researchers will stimulate the neural circuits in order to alleviate psychiatric patients from their symptoms. So optogenetics, we were first introduced to this through simulating predatory switches in mice. So similarly, we could use light to simulate certain parts of the brain in order to have certain behaviors displayed. And nextly, positron emission tomography. This is also known as the PET scans. And tracking certain proteins with the radio tracers, we can see how it flows throughout the body and affects different processes. Deep brain stimulation has a device implanted in patients, and it sends electricity into abnormal parts of tissue, and it would reduce symptoms of damaged circuits. Both transcranial direct current stimulation and electroconvulsive therapy send electricity to the brain in order to help fix damaged neurons. And our last one, also known as DREADDS. So within neurons, certain proteins can be stimulated by different synthetic chemicals. So what researchers are doing are they're sending these chemicals in order to try to help fix these damaged neurons. So what we are proposing is that because certain parts of the brain are more understood than others, for instance, because Parkinson's and Alzheimer's are so common, there's more research done on those areas than others. So we would like to focus on the entire brain and try to map out the entire neural circuit system using fluorescent microscopy techniques. So we could tag certain brain proteins, such as using quantum dots, green fluorescent proteins, and et cetera, to help track where these neurons go and how they affect processes. So for instance, quantum dots, by stimulating them with lights, the electrons can be excited in order to help fix damaged neurons. And similarly, green fluorescent proteins, when excited by lights, they can help neurons generate action potentials. And by understanding the neural circuit system, we would see how certain areas of the brain are connected to others and how certain processes affect others, and we might potentially find something that we might not have considered before. And moreover, we can also use thin slices of mice brain in order to visually see these neurons under these microscopes, because it has been shown that using mice to study human brains has been effective.
Okay, so upon doing this proposal, we might encounter some challenges, such as not being able to see the whole brain as a whole, and this would, not in complete detail, so this would be a challenge because we wouldn't be able to see how everything works in conjunction with each other. Also, confocal microscopes cannot achieve high-speed 2D imaging, and so this would cause the process to be very time-consuming, and results would come in long segments. Spatial resolution is any point source of light that's smaller than the light diffraction, and when this occurs, there's a blur in the image, and so this can cause inaccurate images and not as detailed images, which would cause the results to be not as entirely detailed. And neuronal networks are dense 3D tissues, and because they're so packed and complex, it can be kind of difficult to study them and see what their structure is really like.
Also, another challenge is observing a live specimen, it can go against some people's ethics, and so this would cause problems. Delivering light to the brain is also a problem by putting GFPs and quantum dots in, because they can react with the retina cells before going to the brain. So essentially, our purpose is to map out the entire neural circuit in order to have a better understanding of how neurons have an effect on how we behave and disorders and diseases people have. So by learning more about these different regions, we can pinpoint how different locations have an influence on how people react. And for future direction, we can develop better technology by carrying out this purpose in order to avoid the obstacles mentioned before. And this would not only help this research, but also future research or other current problems. And of course, we can break frontiers by discovering more about the brain.
So these are our contributions and our references, and we are open to questions. Yeah.
Do you have any specific diseases and associated specific areas that you think are understudied and that you would want to target first?
So dementia is a broad, it's a very complex disease, and it's also a very broad topic. So I think because it's so complex, there isn't as much research being done on it as potentially other diseases. So I guess that would be somewhere we could focus first. Okay, yeah?
So you are proposing to map out all the connections, right, in the brain. So it's a very valid proposal. It's actually going on right now as we speak in different places. So let's call it the connectome. So you are talking about fluorescent microscopy, but we know that it's really hard to image deep in the brain, right? So from everything we talked about and everything you guys have learned during these 5 days, how would you actually address that challenge of really imaging throughout an entire brain, that being like a smaller brain, like a mouse brain, or a bigger brain, like a human brain?
We might be able to use like cryo-electron microscopy, because like the electrons can like pass through the sample and possibly see deeper into the sample than just fluorescent microscopy.
And also earlier this week, we kind of looked, as mentioned by other groups, we looked at PET scans, which allows us to trace radio tracers throughout the body. So I feel like that would be very effective in studying how the brain flows and other areas like that.
So if you were to use electron microscopy, I think you will still have to somehow reduce the size of the brain, right? It's still a bit too big. So have you heard anything about how you could process your sample in order to do meaningful electron microscopy images on it?
So I think if we use electron microscopy, it would not, it couldn't be a live brain. It would have to be, it would have to be like, as we mentioned, slices of the mice brain. So I think in order to better study and prepare those neurons within those brains, as mentioned before, we have to freeze it somehow so that during the process of shooting electron beams, we won't damage any of those cells and potentially make an error within our research.
So you will have to slice the brains,
Yes.
correct?
Yeah.
So can you think about another method we talked about where you would not necessarily need to slice your brain?
So this is the same question that I asked the last two groups, right? So we're going to answer it at this point probably if you guys don't have it, so we don't have to keep asking.
So if you were to use fluorescence microscopy and you are facing that quite large object that is a brain, what have you seen, what have we talked about that can kind of help you out by
Yes.
So what I was asking 2 groups ago was multi-photon deep tissue imaging, which I know you guys heard about on Tuesday. And then this particular example is clarity, clarified tissues using light sheet microscopes, right, which you guys heard about, which is where you can scan large volumes very fast to get image maps, physical structural maps of the entire brain. So we won't have to ask it again, hopefully. But for those of you who think about it, well, the next groups, right? We learned a lot about a ton of stuff, right? And I know that was probably a small section of what you learned about on Tuesday, right? But the really advanced techniques, right, are really addressing some of these issues, making tissues basically transparent so that light can go through them, right? Or using advanced multiple photon laser combinations to get deeper into opaque samples. And that was the type of stuff that we were sort of asking about. Like for, I think, your group, when I was asking about the gut microbiome, right, you can image deep tissue imaging in live rats using multi-photon fluorescence microscopy, right? And it's a live organism.
So, since you are interested in the subject, you should know that there is a multi-million-dollar institute in Seattle that is called the Allen Institute, for which the sole purpose is just to map all of these connections. Okay, so this is a very relevant topic of research. And one of the approach that we are doing is to tag all of these neurons with fluorescent proteins, like you mentioned, okay? And combining fluorescent microscopy and electron microscopy together. So, and it's a very neat approach, very powerful, generates huge amount of data, which at this point we don't even know how to handle. It's so huge. But the idea is to really take an optical image first, then an electron microscopy image second, and then slice the brain under the microscope and start all over again like this. So it's a serial sectioning under the microscope. Very powerful. Takes a lot of time though.
All right, thank you, ladies. All right, so we are about halfway through presentations at this point. So let's take a little bit of time to stretch, use the restroom if you need to. So let's take a 10-minute break, and let's be back here in 10 minutes.
It's useful, right? But
9 or 10? I think we were the last ones. I think so. Let me. is this one. All right, so if everyone can please take their seats, we're going to go ahead and
So, oh wait, I'll wait for a second. All right, so. Good morning, my name is Pritahir. I'm from Warren High School in Downey, California.
I'm Nathan On, and I'm from Palos Verdes Peninsula High School in Palos Verdes, California.
My name is Bryant Liu. I'm from Fluorescence Preparatory School in California, and today we'll be talking about optogenetics and their applications to colorblindness. All right, the abstract. Before we dive deeper into this, can I ask, who knows what optogenetics is? Raise your hand. Oh, that's quite a few of you. Well, for the people that don't know, optogenetics is basically a technique where scientists shine blue light onto the species animal's brain, in which they could trigger natural behaviors such as aggression or motor functions like moving the arm. Now today we'll be applying optogenetics to potentially curing color blindness, in which we will be shining light onto the occipital lobe of the brain, and which will trigger the cones in the eyes to receive more light.
So let me give you guys some social context and a little bit of background about how our eyes perceive color. So, or first, hold on, let me start with the question. What percentage of males and females do you think have color blindness? Just out of curiosity. Like, or what percentage males and then what percentage females?
I have a guess. I would say 30% of males and maybe like 5 to 10% of females.
Nope. No? Anybody else?
Straight denial, man.
60 male, 45 female.
Nope.
Not even close.
One more. That's really close, but no. Actually, it's 8% males and less than 1% for females. The reason why is because DNA, or so males have X and Y chromosomes, and females have X and X chromosomes. So the chromosome that carries this trait is on the X. So because females have two X's, that it gets transferred over to males. It has a higher chance of getting transferred over to males than females. Okay, so color blindness is when people, so they're, I don't know where, okay, so there's different, okay, much better.
Yeah. Yeah.
This week was that there's two different receptors for color. People have rods and cones that perceive light and color. So rods allow us to perceive black and white. So I guess, they allow us to perceive black and white. And then when we have, cones are what allow us to perceive color. That's the part that we want to try to fix. Because cones allow different amounts of light into the eye to help perceive color. So when people's eyes, I guess, don't let in enough light in a sense, they get different variations, such as that. So that's why there's different types of color deficiency. So there's like tritanopia. Yeah, you guys get the point. So essentially, that's what causes that. And then this affects a lot of people around the world, like 8% of males around the world. That's a huge number. So if we were able to cure this, we would essentially be allowing numerous people to get, or we open their eyes to a whole new world of color. Like,
literally.
Literally. Imagine somebody wants to become an electrical engineer, but then they can't because they can't tell the difference between a red and a green wire. Stuff like that. So that's part of the reason why we wanted to research this a bit more.
So the main question of this is to find out what part of the brain controls the cones, which is primarily responsible for color vision in the eye. And these receptors also allow insight of the mapping of the brain, which is a huge concept of the Brain Initiative. And their main mission or goal is to completely understand the brain.
All right, precedents. So there are numerous amount of scientists that are also researching using optogenetics, such as here at CNSI, Dr. Katsushi Arisaka, in which he used, like the example before, he used optogenetics to trigger aggression in mice. I wanted to show a GIF, is that how you pronounce it? I don't know. And basically, what he did was he put a stick with mice in it. And when the blue light is not turned on, the mice do not attack the stick. However, when the blue light was turned on, the mouse attacked the stick, in which proves that optogenetics did trigger some sort of aggressive behavior in the mouse. All right, and here's our project proposal. So our team will use optogenetics to try to reduce or remove colorblindness from the person affected, and we will do so by first applying anesthesia to a live human patient, because we can't really do this with mice because they can't give us a response on whether or not they see color or not. They'd probably be like, squeak, and when they don't see color, and a higher pitcher squeak when they do see the colors that they don't see. So I think the only way we could do this is probably with humans, in which we actually develop a language that can actually help us communicate with each other. So first we'll be applying anesthesia or not, depending on the patient and what they prefer. And we will be probing, we will be using optical microscopes, a mountain optical microscope, to see some places in the brain, and we will probe some areas part of the occipital lobe, which is the lobe responsible for vision. And so basically, we'll just keep probing and probing until they say, like, whoa, color. And basically, when they do say that they see color, we will use fluorescence microscopy, in which we will inject some fluorophore or GFP into that area of the brain, and that will react to the neurons inside the occipital lobe, and we will see. What do my notes say? And we will shine blue light in that area to see where the GFP is, and we could apply it to. If we find the right neuron in which they reacted to, we could potentially change the future of neuroscience and add more to the mapping of the brain, which is, oh, sorry to mention, this topic's related to the Brain Initiative, in which they're responsible for, our mission is to map the brain.
So people with normal color vision are called trichromats, and people with the faulty color vision they're known as anomalous trichromats. And the main confusion is that once people experience color blindness, they don't really know what type, because there are many broad types of color blindness. But there are three main types. There is the proteinomia, which is a reduced sensitivity to specific red light. And there is deuter anomaly, which is the sensitivity to green light, and it's also the most common type of color blindness. And then also there is the trite anomaly, which is a reduced sensitivity to blue light, and is the extremely rare case and is the most uncommon case of color blindness. So here is more charts on the specifics of the three types of the color blindness, on where people can perceive different types of color for the normal color vision. So in this photo, the top left picture is someone with the normal color vision, and the top right is someone who has tritonopia, which is a reduced sensitivity to the blue light, which is also the most uncommon case. The bottom left is the normal color vision, and the bottom right is the deuter anomaly, which is the reduced sensitivity to the green light, but also the most common case of color blindness.
So discussion. So to summarize, essentially, or based on the experiment that we saw on like the first day of class, we want to see if we're able to trigger, or so the blue light was able in the video we saw in which the mouse has blue light shining on its mind. it triggers a sense of aggression in it. So we want to see if we're able to trigger something that'll cause the cones in the retina to, in a sense, like perceive more light to help people perceive color. And however, or as one of the issues is that we may accidentally like randomly trigger the wrong thing, as in like the mouse. We don't want to trigger aggression in people, but that may be an issue we may run into while testing it. But however, once we conduct those tests, obviously we hope to release it to, or we would hope to bring it to hospitals and so forth to help cure color blindness.
These are our contributions, and these are our references. Thank you very much.
Any questions? Oh, wait, no, after the questions, after the questions. Questions? Oh, wait, actually, so I want to explain that picture. We actually were in one of the labs, and we wanted to see the fish that they were working on, and I managed to take a picture of it, a video of it actually, and that one came out really cool, so we thought, why not show you guys? But yeah.
And this is thanks to Dr. Bento Leila for letting us see the fish in the microscope.
Yeah. So any questions?
First of all, okay, thank you. Okay, first of all, you guys are so hilarious when you are presenting. It's perfect. Like, it really helps. It really helps bring some character to what you guys are presenting.
Thank you.
Thank you.
What I wanted to ask, how is using this optogenetics going to affect the cones that are related to color vision? Like, is there some way that that using this light is going to, like, are the cones damaged or something? Is like, what's the reason behind color blindness, and how is using this going to help?
Do you want me to go? Okay, so essentially so the eye is able to see because it lets in light and so forth, right? So what happens in the cones is that if it doesn't receive enough light, because like it's kind of like you're shining light through a hole, and then you have receptors on the back that will decode it in a sense. That's like a rundown version. But with the issue with the cones is that they're not letting in enough light, or in some instances they're letting in too much light. So we want to see if we're able to, in a sense, like kind of, or you know how cameras have apertures, how you can open it and close it? We want to see if it's something like that. Because if it is, then that could potentially solve our problem. Yeah. Any more questions?
So aren't the rods and cones on the retina in the eye, not in the occipital lobe? So why would you like use a microscope for an occipital lobe?
Okay, so basically, you use a microscope for the occipital lobe to locate the neurons in which you're probing, which could possibly be connected to the cones. And so when you probe the occipital lobe and it triggers the cones, we would use the microscope to locate that specific neuron, and we would inject the GFP to locate that neuron, and we would provide a cure for color blindness for that specific person.
And then the occipital lobe is responsible for vision for the brain.
Does that answer your question? Okay.
We have time for one last question, if there is one.
So
Yes?
I think, I mean, I get the gist of what you're trying to do, but I'm trying to understand where the microscope comes into play, right? I mean, at the end of the day, it sounds to me like you need to genetically modify the neurons in the occipital lobe of the brain, and then you're going to try to figure out where to pulse them with blue light to manipulate the cones in the retina, basically, right? So what do you need to visualize to do that? I mean, you're going to try to mount a microscope onto the head of some human or some animal, right? What are you looking for, other than the fact that you have basically genetically modified neurons in that region that you can stimulate with light?
Oh, sorry, I didn't clarify. Is the doctor or the person using the probes on the occipital lobe that'll be wearing the microscopes, so they could locate that specific neuron?
So you think that there's some specific neuron or set of neurons in that region of the brain
Yeah.
Yes.
And you need to figure out what it is, where it is, so that you can.
Essentially, yeah.
Yeah, that's where the GFP comes into play.
Yeah.
So, which one is yours?
It's the Angelina one. Which one do you want to take? Microphones. Which microphone do you want to take? That's fine. I don't mind going behind
Hi, everyone. My name is Angelina Cheng, and I go to Whitney High School.
I'm Christina Zhao, and I go to St. Margaret's Episcopal School.
Hi, I'm Elise Kawai, and I attend Kaimaki Christian School.
And today we're going to be talking about using microbiomes to treat lung cancer. So, within the human microbiome, the majority of the microbial cells reside in the gut. So, because we know there's a correlation between these gut microbes and the overall body's health, we believe that there is a possible cure for lung cancer through the manipulation of these certain gut microbes.
So, what are microbiomes? Microbiome is a community of microbes that inhabit in a particular environment. So, there are many types of microbiomes, which some examples are bacteria, viruses, and eukaryotic microbes. So they're really capable of living in harsh conditions, which they can stand extremely cold or extremely hot conditions.
So there are two main topics we're going to be discussing throughout this presentation. The first one is microbiome, as she mentioned before. So it's defined as a community of microbes that inhabit a particular environment. And the other one is lung cancer, because lung cancer is one of the most hard to diagnose because it's unlike other ones because it's within your body. You can't really tell because it's underneath your skin and within an organ, which is why over 1.69 million deaths worldwide in 2015 occurred. And so far in the United States alone, this year, over 234,000 people died. And our topic we're trying to answer today is, by manipulating specific microbes in the gut, are development in the lung cancer treatment possible?
Depending on its success rate, the proposed treatment may reduce or provide an alternative to traditional radiation and chemotherapy treatments, both of which share severe, sometimes permanent effects. These side effects can include fatigue, nerve damage, increased chance of infection, and tissue damage. However, since some microbiomes can be affected by targeted medicine, as we learned this week, this form of treatment may have less harmful side effects. Consequently, there will be a higher quality of life for patients in addition to longer lives. Moreover, should this form of treatment work, a similar approach may be possible with other cancers. In November 2016, University of Texas, Dr. Jennifer Wargo studied over 100 fecal samples from people with advanced skin cancer. She ultimately discovered that patients with more diverse types of bacteria in their gut responded better to immunotherapy treatments, thus supporting the hypothesis that gut microbes have a noticeable effect on health. This is backed by another experiment in which microbial-treated mice who were injected with liver cancer had 40% smaller, as well as less heavy tumors than their untreated brethren. Moreover, nanoscience is becoming increasingly integrated in medicine. This year alone, there are currently over 80 active or recruiting clinical trials for cancer nanotechnologies. In order to develop a means to fight and perhaps even cure lung cancer through the manipulation of gut microbes, we will need to research first, as relatively little is known about these microbes. Similar to Dr. Margo's approach, fecal samples can be collected from patients with lung cancer and analyzed with confocal microscopes, a form of nanoscale microscopy. If more resolution is needed for our samples, we are considering using atomic force microscopes since it also does not damage biological samples. After analyzing the data pool, we can determine which microbes respectively help the body resist or succumb to lung cancer. Two separate medicines can then possibly be developed to stimulate the former and to kill the latter. We could potentially use bacterial contractile nanotubes, which are highly precise in our cure. Additionally, this process might give us more understanding to how the gut microbiomes affect health and lead to the identification of more microbes.
So some of the challenges we might be meeting is that to create the data to extract the data from the microscopes and stuff, it takes a very lengthy time to analyze the data also, and to be able to have the confocal and the atomic force microscopes. It costs a lot of money, so it's kind of it just costs a lot. And so as living humans are part of this study, there's potential for other factors to corrupt the data. So due to some facts that are really we don't really know a lot about it, so some inferences and interpretations will be made. And so for our proposal, one would be we get to study the microbes in relationship to lung cancer, and two, from the data gathered, we will develop a new treatment for lung cancer. So basically, the second goal might not be reached, but if it do happen to get reached, it could be a new treatment for lung cancer, or it could work with some kind of medicine that already exists today. So work together to cure lung cancer. So further information can be researched and used for future studies and research.
Angelina and Christina jointly developed slides 3 and 8. Angelina edited slides 1, 2, 4, and 10, while Christina edited slides 1 through 4 and 8 through 10. I came up with the proposal idea and did the development research for that, as well as created and edited all the slides except for slides 3 and 8. Christina and Angelina jointly used 3 out of the 19 entries on the works cited list, and I used the remaining 16. This is our works cited list. Thank you for coming to our presentation, and thank you for listening.
So do you guys have any questions about anything?
Can you guys clarify a little bit more about what information you guys would want to gain from your system by using atomic force microscopy with the two cell types, I guess your lung cancer and the microbiome cells? What information are you trying to access with the technique of atomic force microscopy?
We were hoping to access exactly what microbes do affect, or perhaps do affect lung cancer, whether they help the body resist lung cancer, or whether they accelerate the effects of lung cancers.
Any other questions?
I have a question. So in trying to identify different parts, positive contributors versus negative contributors in the microbiome for prognosis with lung cancer, how would you try to differentiate or identify them?
I think that we would try to differentiate them and identify them by also keeping aware of the prognosis of the individuals from which we are asking for samples. In Dr. Wargo's experiment, from which we were inspired for this specific or our own idea, she kept track of the prognosis, and then using the samples, the people who who all had more diverse bacteria, for example, were the healthier, they responded better to treatment, while the people without, so we would be looking for trends.
How did she measure healthy bacteria versus not? Just total amount? Or?
I did not have access to her full study at the time, although I did check to make sure that the source that I found her results was reliable.
Any other questions?
So you mentioned that the experiments that you would be doing are somewhat controversial. So for the people who are kind of like, why are you doing this? This is not a good thing. What would you say about the negatives? Like, why do you think that the positives outweigh the negatives here?
I realize that while it can be time-consuming, I do think that nanoscale microscopy has a lot to offer in the realm of medicine, and that there are in fact already drugs that have been developed from nanoscale science. So I do think that also we talked about at a point in science where there are certain limits to every single approach, but I think that in addressing certain limits that we have now in medicine, that nanoscience can help fill in a lot of the gaps.
There's no more questions, then thank you very much, ladies.
It's number 8. It's at the end of the earlier list, I think. Right there.
Yeah.
Okay, wait.
All right.
I don't know how to use Mac, so you better. You press the arrows.
Just press the button. All right. Hi, everyone. So we're presenting about how bacteriophages can be used to change microbiomes. So my name is Joey Zhu. I go to California High School in the Bay Area.
My name's Jake Penuelis, and I go to Camarillo High School.
I'm Trevor Sargent, and I go to Maricosta. Okay, this is our abstract. The question that we addressed was, what technologies can scientists use to accomplish this difficult task that we explained in the topic slide of reshaping microbiomes and ultimately getting rid of harmful antibacterial resistant bacteria. Bacteriophages, if you're not aware of what they are, they're basically viruses that infect bacteria with their own DNA. They're the next, harnessing what they can do, they are the next big step towards curing very resistant bacteria like carbapenem resistant Enterobacteria acacia and cancer. We plan to modify proteins on the bacteriophages to make them bind to bacteria of our choosing, therefore curing the disease.
So for the topic slide, a microbiome is a group of microorganisms in a particular environment, and biophages are a virus that parasitizes a bacterium by infecting and reproducing inside of it. So microbiomes are unique to everyone. They're not specific, like me and Trevor will have completely different microbiomes within us, and they can influence our health. So knowing which bacteria affect our health in which ways can we might be able to modify these microbiomes and potentially get rid of any destructive cells or improve any others that are not working to their full capacity.
All right, this is the social context. The bacteria have been evolving and become a lot stronger after the almost overuse of antibiotics. This includes bacterial infections such as CRE, which was mentioned in two slides ago. If we use bacteriophages to change the physical makeup of harmful bacteria, billions of people in the future can live through diseases that were deemed incurable in the past. Also, there's no shortage of bacteriophages on the planet. We have more bacteriophages in our system than actual bacteria. Yeah.
So for our precedents, scientists are currently genetically modifying viruses to cause antibiotic-resistant bacteria to kill themselves. They're also studying how different diets and activities can change our microbiomes. It's been shown that they can transplant microbiomes from one subject to another to change the health or the personality of the original subject.
All right, now a project proposal. We can use quantum dots and fluorescent proteins to identify harmful bacteria, modify the receptor binding proteins of the bacteriophage to go to unique protein sites on the individual on the bacterial on the colonies, and heal or destroy the cells depending on whether the bacteria are harmful or damaged.
So basically, like all the other projects and proposals mentioned earlier, this one is going to have challenges if we want to actually try to reach our goal. First of all, we are dealing with a microbiome which is many different species of not just bacteria but other animal cells, other protists, etc. So the protein research to find all the different proteins on each cell membrane is going to need a lot of genome sequencing. And although this kind of genome sequencing process has been getting very fast recently, but today it's still very costly and time-consuming. Also, just like bacteria can develop resistance to antibiotics and become superbugs, there's actually evidence that bacteria can develop the same kind of resistance pattern to bacteriophages. The problem is that proteins on different cell membranes can actually all be the same. So if we're not careful, then maybe we find the wrong protein, or maybe we find a kind of receptor protein that also binds to other helpful bacteria. Then the bacteriophage protein will actually start to attack helpful bacteria in the human microbiome and start disrupting that delicate ecosystem. In addition, there also might be some toxic, harmful byproducts in the lytic cycle as bacteriophages attack populations of the target bacterium. So, the scientific community kind of can be seen as like is kind of like a tower. We keep building up higher, but then the people who are building up right now have to have kind of, as the saying goes, stood on the shoulders of giants. So, following this, we have to use a lot of microscopy techniques, like for example, as they mentioned earlier, fluorescent microscopy. And then also, in addition, we will also have to use other techniques for like electron microscopy and scanning microscopy to actually deal with the proteins themselves and image them at a very high resolution. Our research in fluorescent microscopy will also be beneficial to other future biologists. They deal with a large amount of raw data today, but they can't make much sense of it as it's like mostly all numbers and quantitative observations. However, fluorescent microscopy is very special in the way that you can use it to color code, kind of look at interactions, find much more meaningful qualitative observations. So then scientists can use these to quickly draw models for their own projects and kind of use that data as a complement for whatever they're researching. So these scientists can also expand our observations to research similar bacteriophages and diseases and also possibly find treatments for them. Ultimately, at the very end, we can even kind of like put all this data together with other scientists kind of as a jigsaw puzzle to find general patterns or behaviors in human microbiomes in general.
So here's our contributions page and our references.
So right now we're open to questions. Barney?
Do you guys guys say this. Do you guys fear that because we keep engineering these bacteriophages, is it possible that the microorganisms and the microbiomes could eventually adapt and be resistant to these bacteriophages?
So, as we kind of mentioned earlier, this is true because the process of natural selection is going to yield several bacteriophage-resistant bacteria. However, actually one of the challenges that scientists predicted is that bacteria will actually develop resistance to both antibiotics and bacteriophages. However, in most cases, when they develop resistance to one, they actually sacrifice their resistance to another. So then you can actually use these bacteriophages and antibiotics in tandem to treat certain diseases.
And then also, if it creates new protein sites, we can identify the uniqueness in each of the new bacteria, and then we can modify our bacteriophages to then bind to those sites instead.
Any other questions?
Robert.
Go ahead, and then I'll ask.
So how, I think, well, maybe, what's the kind of precaution or safety, you grant the safety of this thing, but if you want to use this to treat diseases, maybe kill bacteria, how can you make sure that when people use it, it doesn't kill other good stuff?
Well, each bacteria has binding proteins that the biophage will link to. So we just make sure that we find a unique protein that our biophage will only connect to the bad bacteria that we are trying to isolate. So any of the good bacteria will not have that binding protein, and the biophages will not inject their DNA into them.
Yeah, so that's why it's important that we have to kind of carefully sequence and research every organism in every species within the microbiome so we don't accidentally make a mistake and tip the balance of the ecosystem.
Can you think about an experiment, how you would test for specificity?
For example, we would kind of work with different quantum dots, maybe work with different kinds of fluorescent proteins. We would see that, we would try and kind of see what different indicators will bond to different protein receptors. And then using the information that we already know about those indicators, like the quantum dots, then we could deduce what proteins are actually comprise the bacteria of interest. And this can also be confirmed again, with genome sequencing.
And microbiomes are kind of found everywhere. So we could use this experiment in mice or other animals to test how this will work.
So maybe you're right, you would take the genome, try to identify all the different proteins you expect to be there, and then utilize fluorescent tagging of that specific protein to verify that it's actually there on the bacteria you want to kill, and then those would then become your target, and you would use that protein.
So basically, this kind of experiment design is multifaceted. It eliminates a lot of possibility of error.
Yeah, I agree. You'd have a lot of error if you just went for the genetics alone and then tried to see what was really on the outside of the cell. You'll find they have the capacity to make lots of proteins, but whether they do make them or not is a big variable. So verifying that with fluorescence would be a good idea.
Yeah.
Any other questions? Okay.
I did have one, but Laurent actually asked it for me. So, or I mean, I could still ask it. So I guess it's a related question. So, so you guys mentioned that you would use fluorescence tagging to find the harmful bacteria. What specifically would you be looking for in the harmful bacteria? So how would you actually tag only the harmful bacteria and not the good bacteria?
So the thing is that we actually have to so again, we have to use a lot of different methods to tag the bacteria. Maybe we first kind of just test a lot of different indicators, kind of try to see which, let's say we use a green fluorescent protein, and then we also use quantum dots which fluoresce with a red color. So if those happen to each tag to different bacteria, let's say the red one happens to bind with inert or inert or native bacteria, and then the green one binds to the harmful bacteria. We could kind of see that like all the green specks under the microscope, maybe they have like very aggressive behavior, maybe they quickly divide or take up resources. So we could kind of use that to deduce which bacteria don't belong in the microbiome.
And once we determine that, then we can map out that one of the strain of bacterium, and then we can find which protein sites are unique to just that bacteria, and then we can send in the quantum dots to make sure that our hypothesis is correct, and then bacteriophages go in.
So if I understand you correctly, do you mean to say that you would be monitoring the behavior of the cells to try to determine which is harmful and which is not?
Basically, we're just trying to look for as much evidence like from all around the board as we can, so then we could kind of draw a more general or unbiased conclusion.
This is why we would also kind of test it in mice or something first, so we could see which microbiomes are harmful.
But in mammals, not other animals, because they relate most to us, and if we go into like amphibians or like birds, then it's going to be a lot different than what would be in our genome.
So given that there's so many strains of bacteria, and it's a massive project to figure out which ones might be good and which ones might be bad, where do you think you could turn as a source for information on that, so you don't have to do it all yourself?
So as I was saying, it's kind of like the scientific community is kind of like a tower. Like maybe just below us, there are scientists who are working to improve the genome sequencing process, or maybe there are already databases for like the said bacteria.
Great example. And you could come up with panels, 20 example harmful pathogens and 20 example inharmful pathogens, and start with that.
Mhm.
And as we get more and more metadata, you have more and more to work with your techniques to feed off of.
Mhm.
Okay, great.
Yes.
All right. Thank you, guys.
So you guys had yours.
We're grouped into 9.
Oh, right there.
Hey. Hi guys, my name's Arjananda, and I'm go to Concord Carlisle High School in Concord, Massachusetts.
I'm Madison Toomey. I go to La Jolla Country Day School in San Diego.
I'm Megan Gramling, and I go to Laguna Hills High School.
And today we're going to be talking about the permafrost microbiome.
So this is our abstract. I'm not going to read the whole thing because we're going to explain it in the presentation. But basically, our project deals with permafrost, which is chronically frozen ground, and how it is thawing due to increased global temperatures because of climate change. And this is causing increased release of methane that's trapped in the permafrost. And the goal of our experiment is to see if the methanotropic bacteria, which is bacteria that consumes methane for energy, can decrease the amount of methane being emitted into the atmosphere.
So before I get into the main concept and topic, I'm going to define what a microbiome is. We talked about 2 definitions, the first being all the microorganisms that make up a particular environment, and the second being the genetic makeup of all those organisms. And now I'm going to define the permafrost, going off of what Maddie said. It's chronically frozen land that for 2 or more consecutive years that includes soil, gravel, and sand within frozen ice. And if you hadn't guessed it yet, they're most common in the northern locations like Canada, Alaska, Russia, and more. And so our main question is, how can we change the structure of the permafrost microbiome in order to reduce the emission of methane? And so what's happening right now is, due to the rising global temperatures, the permafrost ground is beginning to thaw. And then because of this, the plants in the microbiome are releasing carbon, and the archaea within the microbiome are taking in the carbon and then releasing methane.
Okay, so for the social context, carbon dioxide and methane are both greenhouse gases. However, methane is much more harmful to the environment, and the increased release of methane into the atmosphere will raise global temperatures even more, which will in return increase the melting rate of permafrost and release even more methane into Earth's atmosphere. This will essentially trigger an irreversible cycle that will have detrimental effects upon our environment. It is extremely important to address this issue because although people have calculated the potential progression and outcomes of climate change, most of these figures don't account for permafrost thawing, and this will rapidly accelerate the timeline of global climate change. Overall, decreasing the release of methane into Earth's atmosphere can help further decrease climate change, and this proposal could benefit humans in the aspect that global warming could become less of a problem for future generations.
Okay, so now I'm going to talk a little bit about the precedents and some studies that have been done about this topic already. So far, scientists that are studying methane emissions from permafrost have been trying to answer questions such as how much carbon is actually under the permafrost, how fast the methane is being emitted, and what impacts it can have on climate change. And so one of the leading scientists studying this issue, David McGuire, who's pictured right here, has estimated that the permafrost contains about twice as much carbon as there is in the atmosphere. And much of that carbon is frozen into the permafrost, so when this carbon is emitted into the atmosphere as methane gas, the effects could be extremely harmful to the environment. And so this image on the screen shows the study sites of permafrost completed by many scientists, mostly in Alaska. And this just proves how much permafrost is in one area of the planet.
So this is our project proposal, and our solution would be to introduce methanotrophs to control the methane emissions. So now I'm going to talk about what they are. So they're bacteria that live in extreme environments, in areas with limited oxygen, with high methane levels, and limited concentrations of nitrogen and copper. These conditions are really similar. They take in methane as energy. They break it down. And the conditions I just described are very similar to that of the permafrost, so they could survive in those conditions as well. They oxidize CH4 methane using an enzyme called monooxygenase, whose essential cofactor is, in fact, copper. So now I'm going to walk through the project proposal. So what we're going to do is we're going to first simulate permafrost conditions in the lab, and then we're going to slowly raise the temperature of that simulation, and this is without the methanotrophs, and then gather the amount of methane that has been released. And then we're going to do another simulation using an identical permafrost simulation, and we're going to introduce the methanotrophs into this beforehand, and then after raising the temperatures, we're going to see how much methane has been released, if any, and determine the effect that the methanotrophs have on combating the methane emissions.
Okay, so moving on to the challenges. One of the biggest challenges for our proposal is that we're unaware of the amount of copper that's within the permafrost environment, and copper plays an important role in assisting the enzyme monooxygenase deteriorate the methane, and it's necessary to react. Another challenge is human interaction with territories containing permafrost. Causes the permafrost to thaw even quicker and release methane and carbon dioxide at a much faster rate. The image on the way right shows homes in Siberia on top of permafrost, and the human interaction with the permafrost forces it to thaw rapidly and collapse some of the permafrost and results in a shift of the land. And then lastly, a third challenge is obtaining enough methanotrophs to properly execute this experiment because there is a limited number available. And then also the last challenge is actually implementing the methanotrophs into the permafrost environment.
Okay, so for the time being, we suggest solely testing the methanotrophs on methane in soil in artificial permafrost in the labs to determine the effectiveness and impacts that this proposal could have on the environment. And we want to utilize the electron microscope to analyze a sample of methane that would be frozen with cryo-EM to simulate a similar extreme temperature that the methane experiences within permafrost. And next, with a combination of methanotrophs and copper, we would place a small sample of the methanotrop copper mixture on the methane sample to witness the reaction between the variables. So if this works, we will incorporate the methanotrophs into the actual permafrost microbiome, which will hopefully reduce the overall methane emission that comes from the melting permafrost.
These are contributions and our references and sources, and that's it. Any questions?
Why is methane bad for the environment?
Do you want to go for it?
So methane is a greenhouse gas, and when it's emitted, it goes into the atmosphere, and so like usually the atmosphere insulates the planet, so it like traps the sun's rays to keep us like warm and to regulate the temperature. But if there's too many greenhouse gases that could in the atmosphere, that could affect the temperatures, which is what we're seeing with climate change right now.
I'm not quite sure what you are intending to measure with the electron microscope. Can you elaborate?
So we're gonna, in the lab, we're gonna take a sample of the first simulation and sort of like freeze it or get it to the temperature, and using cryo-EM and see the makeup of all the microorganisms, and then we're gonna take the sample from the second simulation with the introduced bacteria, and we're gonna see if there are any like detrimental impacts of introducing the bacteria and how like it really affects all the other microorganisms in the microbiome as well.
So two different conditions.
Yes.
Observed under the same.
So like a control and the experimental group.
So, do you know what happens with the methane when the methanotroph consumes it? Do you know what products it produces?
Do you know? So what would happen is it releases carbon dioxide, but then that would cycle back into the, which you know is a greenhouse gas, but that would cycle back into the archaea, and it would break down. So it would kind of be just like a big cycle of breaking down gas, and then another organism would take it in, and then it would release and break in. So it's kind of just like a new cycle.
Okay, so, but it eventually kind of, it stops like that carbon dioxide from becoming a greenhouse gas itself, right?
Yeah, the archaea would break it down.
Okay, thanks.
For what I think.
Small comment. You showed a map of Alaska
Mhm.
with a lot of colors.
Yeah.
What were those colors?
Those were just like basically just a geographical map showing like the different heights and like the peaks of the permafrost microbiomes. That weren't like, it wasn't like too like complicated or anything. So like,
So it was like almost like a topographic
yeah,
map.
yeah.
Yeah, it was like how much like permafrost is in each area, and like as the colors got like closer to like green and blue, that was where more of like the actual like super deep permafrost is, and then closer to red and orange was less permafrost.
Any more questions?
So I have a question. When you look at your samples, are you going to do anything in particular to try to mark out your modified cells versus the natural cells of the microbiome in your samples? How will you identify whether it just looks like there's more bacteria total versus more bacteria from your type versus there's only a few bacteria but 100% of them are your modified bacteria, right? How do you tell what's native versus what's your engineered bacteria?
We could, in the second sample, we could introduce like the fluorescent protein to make it apparent which ones are the methanotrophs, so that we're not just like counting all the bacteria.
So then you'd want to use fluorescent microscopy first,
Yes.
I think you're better off tracking copper. I know that you guys didn't probably learn too much about it, but the electron microscope also has the ability to do elemental analysis. Talked about it very briefly on Monday. I showed you an example when I went through EM in about 30 seconds, right, where you can actually use energy dispersive X-ray detection to map out elements, especially metals, right? So you would theoretically see the bacteria that are ingesting and processing or metabolizing carbon would have a localized signature of carbon in that, copper in that region. It would also give you a way to analyze the actual copper content of your samples, right? Whether or not your simulated permafrost maps onto real permafrost, give you a control there, and also to see is it degrading over time. Because there's going to be some finite amount of copper in the environment, even though you have this cycle of the methanotrophs and the arca, right? You're going to run out of copper at some point.
Yeah.
It's another way to monitor and use microscopy to study this sort of stuff.
Thank you, guys. Do you know which file you are? It's Audrey. That one. This one? No.
No. Down. Like in the right there.
Oh, this one?
Yeah.
Hello, my name is Maya Netto, and I'm from Grace M. Davis High School.
My name is Audrey Pettingill, and I'm from Wilsey Wood.
And for the CNSI Nanoscale Microscopy Summer Institute, we will be demonstrating how microbiomes affect the health of living organisms. In this presentation, we are going to show you how microbiomes affect the health of living organisms, and our proposition is to treat bacterial issues in microbiomes by allowing bacteriophages to inject DNA and RNA into infected cells in a process called phage therapy.
So by now, you guys probably already know what microbiomes are, but microbes are like shown in this picture, and they're basically just little microorganisms that cause disease and fermentation, and they're what make up microbiomes. So.
I will be showing you how intestinal microbiomes play a huge role in human health, with several factors such as digesting fiber, brain health, and immune system control.
And I decided to go more into like skin, and so I'm going to be focusing more on acne, which is a disease of the pores. Basically, it just is your glands, your pores, basically up there, have sebaceous glands, and they produce sebum, which is an oil. And sometimes the sebum can produce too much, and then it causes bacteria to get trapped and produces acne. So.
Microbiomes dictate such a wide variety of medical ailments. So if we can harness the power and figure out how to manipulate them, this would result in an array of benefits to society, which would mainly be in bacterial infections and skin diseases.
And one of the like most important or just like really big benefits for acne is like Accutane is a really harsh medicine that you have to take with acne, and so phage therapy wouldn't be as harsh, basically. So and then if you want to go to the pictures. So bacteriophages are shown on the very bottom and over here as well. They're also known as phages, and they are viruses that infect bacteria. They have in the head part, they contain genetic material, and then they have pins and teeth that they use to cling on to prokaryotic cells, which are up there, basically bacteria, and then they inject their DNA. And using phage therapy, scientists go in and either manipulate the genetic material or create a synthetic genetic material, and inject it into
bacteria that is harmful, and then the phages go in and their DNA and RNA take over and then make proteins and then basically just like break the cell wall and infect other cells or other, yeah. Okay.
We are proposing to use bacteriophages as treatment for several bacterial diseases, and the two main advancements we will be focusing on are dermatologic and bacterial ailments. To go about these issues, current research shows that we would try to eliminate the bacterial disease or pathogenesis determinants vectors for bacteria, and by using fluorescent microscopy, one could analyze unhealthy cells and use that information to determine how to construct the necessary bacteriophage needed to treat this particular issue. So in fluorescent microscopy, we could use quantum dots in the phage to show the genetic material and how it would affect the cell we're addressing. If harnessed, the bacteriophages could be applied to medical fields and treat infected cells with healthy RNA and DNA. We could also reverse this process by isolating R-type Fluorescence, which could potentially be used as an antibacterial agent and treat things such as Pseudomonas aeruginosa.
So even though phage therapy has less side effects than antibiotics, there are still like a few issues with the treatment. One of them is that the phages can possibly kill the bacteria too fast. So that would produce too many endotoxins and make the patient sick. Another is it's really expensive. So based on the Phage Therapy Center in Georgia, it costs about $2,500 for outpatient care and then $20,000 for inpatient treatment, not including travel cost, and also if the facility doesn't have your specific phage, it is like extra cost for creating a specific phage for you. Phage therapy also can become resistant, just like antibiotics, and it also, the FDA has not really set up a system for phage therapy, so there's not as much testing done in the US than outside.
Although using these bacteriophages in targeting human health isn't heavily researched at the moment, scientists have gathered information as to the positive and negative effects that may occur to cells and microbiomes when using phages. This information has the potential to lead to a plethora of medical advancements that involve bacteria. In addition to this, new and innovative microscopes are becoming more prevalent. So with this acceleration in nanotechnology and microscopy, it's becoming easier to analyze microorganisms and figure out how they fit together in their collection of genomes.
Yeah, so any questions?
So why do you think phage therapy is more efficient than antibiotics?
So phage therapy, we can like specifically target kind of more than antibiotics. So we're able to like, I don't know, do you want to go more in depth? Like.
So with phage therapy, we're able to customize it to the individual microorganisms in each person. So while antibiotics might not work for everyone, or it may be ineffective to some people, if we customize individual phages for each person, we have a much higher chance of treating the disorder or disease we're working with. Yes?
Since it's like very costly, how would you administer the treatment?
So like how would you take the treatment? There are different like ways kind of, and that's why the FDA doesn't have any systems. Some people take like a concoction, and so it's like almost like a little shot. Others would like, you physically would get a shot, and then there are other like pill forms. So it's just different forms, but there's not a real specific way yet. So it's just kind of like Wild West out there, whatever you want to do.
And part of the fact that it's so costly is because there hasn't been much research in applying phages to the medical field. So if we were able to figure out more how microbiomes react to phages, then we could lower costs and figure out how to revolutionize this technology.
So I was just kind of like worried. So previously you guys said that you said that acne is a disease, and you said that you gotta like take some whatever medicine to like.
Yeah.
But if I have like a pimple, I just pop it. Am I doing? am I
Yeah,
doing
well,
it?
so it can be used for like cystic acne, where it's like, can you pull up the picture? Maybe it's on this slide 5. Yeah. So on the severe, it's like cystic acne. You can't just like pop a cyst. So that would kind of be used in that text for like really severe acne.
That's a relief.
Yeah.
So on that topic, you mentioned Accutane. Are you familiar with some of the other treatments for acne?
Yeah.
What are some of them? Some of those common ones?
Like over-the-counter medicines and
Yeah.
stuff, and like, yeah.
Antibiotics, though, right? Are often
Yeah.
prescribed. It's just
Yeah.
in a severe case, right?
Mhm.
So what are the big advantages of the phage over antibiotics in that case? And in that case, how do you likely think you would deliver the phage?
Yeah, so,
Like we stated earlier, with antibiotics, they don't always work for people because microbiomes are unique in every body. So if we're able to customize phages to people and analyze with fluorescent microscopy how it would affect each individual person's microbiomes, then there's a much higher chance of treating it if like antibiotics don't work for certain people.
So you're saying that antibiotics are less effective?
It depends on the person. They're less customizable to whoever's using them. So if they're not effective on someone, they have a much higher chance using a phage to figure out what's wrong and help treat that.
Yeah.
But how are they effective in terms of eliminating bacteria? Antibiotics?
Antibiotics eliminate bacteria or phages?
Antibiotics.
Do you want to take this one?
'Cause where are most of the microbiome in the body?
In the intestinal region.
In the gut, right? So what happens when you take an antibiotic? It's going to kill all of the bacteria in your entire body,
Yeah.
right? Whereas the phages, part of the big advantage is that they can target the specific bacteria and leave the rest of them alone, right? And in the case of skincare, you also be able to apply it in a way the gut never feels any effect.
Yeah.
So even the phage in the gut might be perfect, or even the bacteria targeted by that phage in the gut where they're not causing a problem might be perfectly safe. So it's good to think about what the comparative advantages and disadvantages of the two ways of killing bacteria is. Right? One's like a nuclear bomb. You've eliminated everything, right? It's like poison. And the other case, it's a selective targeted treatment, right? So you'll have different side effect profiles.
Do you have any idea of how long the phage will survive in the body once it is administered? Have you read anything about it?
Not really, but do you think longer than antibiotics?
I think,
Yeah.
well, no, probably with phage, they're pinpointing the certain problem. So as soon as they treat the cells, it shouldn't be a problem in how long they last in the body because they're already,
Inject their
yeah.
like, DNA and stuff, so,
Well, the point is it's got to get to the cells, right?
yeah.
So there's always this discussion of what the circulation time of any treatment in your body is. Is it going to make its way to where it needs to go before your liver and kidneys get rid of it? I think that the efficiency of phage, which is, I mean, to Michael's point, it's specificity and efficiency. Right? So it only goes to certain cells, and when it gets there, it is incredibly efficient. Like we learned about on Monday from Professor Miller's talk, and you're talking like they are killing machines. This is what they're designed to do. So the topic of both groups is a really great thing. I think you're going to hear a lot more about it.
Any last questions? Our speakers again.
Good job. And good job to all of our presenters today. Let's give everyone a round of applause. Because I'm sure as For parents, I'm sure as you've all seen with the results of all of these presentations, these students learned a heck of a lot in a very, very short period of time. And it's not easy to take in 3 new techniques that usually only get exposed to in upper levels of undergraduate, maybe graduate, even just graduate school, and to learn them all in just a few days and then develop an original idea and present it. It's really, really a monumental task. So I hope that you guys were all as impressed as we were, because everyone did a very good job today. So good job, everyone. And before we break and head to a lovely reception where we can relax and I'll have some food and beverages, I wanted to first bring up each of our 2 instructors that are here today so far to say a couple of words. So Dr. Bentalila, can you please come up and say some words?
So first of all, I would like to congratulate you all, because first of all, you have been a lot of fun to teach. I really love the questions, great questions. You have been engaged. I think we all appreciated the level of your presentation, and I think we hope to see you back essentially, maybe sooner than later. So hope you learned a few things. I would like also to thank the parents, because you gave us your kids for a week. I hope they learned a lot from what they saw here.
Dr. Stieg.
I don't get any That's fine. Yeah, so I'd just like to say congratulations to everyone, all the students. I mean, I don't know if you realize, but the content for this course, we teach, Laurent Hong and I teach in 10 weeks. You did in, not the entirety of it, but in a week, and you got basically dropped into the deep end of some very complicated things that you probably haven't heard of before. And if the presentations are any indication and the questions that we've gotten of, you know, the level of engagement, interest, and how much you've absorbed, I think that it's another example we see every year of some really motivated young, bright minds who are interested in solving problems in the future by leveraging their intellect and creativity. So I'm really happy to see the outcomes of the projects. It shows me that you've obviously integrated a lot of the stuff that we've talked about, and I wish you all the best. Hopefully some of you will see back at UCLA in a couple years, and if not, wherever the future takes you, just keep remembering that, you know, thinking creatively, thinking differently about things, and really pushing the limits are going to help us all in the future. So congratulations, thank you to the parents, and I guess we'll wrap it there and invite everyone to come out into the lobby and enjoy a little reception. Say your goodbyes, or at least your goodbyes for now. Life is a long time, so you'll probably see each other again someday, and we hope to see you too. Thank you.